Chapter 3, Section 7, Principles of Fermentation. Now, fermentation is something that many organisms can do, and it's just under what conditions will they be doing that? For us eukaryotes, many of us eukaryotes, us mammals, we do fermentation, lactic acid fermentation, when oxygen levels in our tissues get low. Other eukaryotes can do it, an alcohol fermentation, or some can do lactic acid fermentation, when oxygen gets low, or, even in the presence of oxygen, they just don't use it. And we'll talk about why in this section and in following sections. Many organisms are just going to do fermentation just as part of their natural metabolism, okay? It's going to be in low to no oxygen levels, and we've just, over the centuries, over the millennia, have come to just utilize them. You want to make beer and wine, alcohol fermentation by yeast. You want to make yogurt and cheese, lactic acid fermentation by certain bacteria. We're also now seeing that fermentation of the organisms in our microbiome can also influence our health. The bacteria you have growing in your large intestine right now. Yeah, well, what are they doing? Which ones of them are fermenting? How are they fermenting? The purpose of fermentation is the oxidation of NADH, basically in a nutshell. NAD is the limiting factor in metabolism right now, as far as we're concerned. There's only so many NADs, just like there are so many ADP, so many AMPs that can be made to ATP. These are limiting factors, which is why we have to go through this whole cellular respiration thing. NAD, since there's only a set number there, normally, under normal conditions, this excited electron would be passed on during the electron transport chain of oxidative phosphorylation. But if there's no oxygen or no final electron acceptor, that electron transport chain shuts down. Oxidative phosphorylation shuts down. Oxidative phosphorylation shuts down, so does the citric acid cycle. The only thing that can still work, glycolysis. But the thing is, for glycolysis to work, there has to be NAD available. So we'll see some organic compound gets turned into pyruvate. So some sugar gets turned into pyruvate. During that process, you get NAD reduced to NADH. Well, all of those gets used up. You get some substrate level, some ATP made during this process. You now have, the final pyruvate. But the problem is everything else is shut down. You've gone through, you've reduced all your NADs, you've, made whatever ATP you can. The cell's still metabolizing, it's using up the ATP, but there's nothing else you can do. Unless you turn the NADH back into NAD, which will allow you to go through glycolysis one more time. Take that oxidized compound, that pyruvate, turn it into lactic acid. By turning it into lactic acid, you oxidize NADH back to NAD. You can go through another round of glycolysis. You take that pyruvate, you turn it into acetaldehyde, which will then become ethanol that's excreted out. You take NADH, you oxidize it back to NAD, and you can go through one more round. So all fermenters have to conserve energy, okay? They have to conserve energy, not only to meet the two laws of thermodynamics, but the fact they have a limited amount of energy, so they can't waste. Metabolism has to slow down. Only certain functions now that are really necessary for the cell have to keep and maintain that redox balance. Who's getting oxidized? Who's getting reduced? Where's the electron coming from? Where's it going? By going through this fermentation step, the cell can go through glycolysis or whatever version of, you know, so that ATP can be synthesized in a substrate level phosphorylation event. Again and again, a few, a few, a few. It's not high energy. There's not a lot there. Think of this as just limping along. You'll still get where you're going, but it's going to take longer and it's going to take more work to get there. Yeast take glucose to make 2-ethanol, because remember, glucose gets split into 2 pyruvates. Each pyruvate gets turned into acetaldehyde, which ultimately becomes ethanol that is excreted out of the cell. The ethanol is a 2-carbon thing, so there's the bubbles in either your beer, your champagne, or the air bubbles trapped in your bread. A couple of ATPs get made from glycolysis. NADs keep getting regenerated. NADH oxidized back to NAD. NAD gets reduced to NADH, oxidized back. And it just keeps going back and back. Well, kind of the same thing here with lactic acid fermentation. Glucose split in half to 2 pyruvates. Pyruvates get turned into lactic acid. Different enzymes here for lactic acid fermentation than what we see here in ethanol fermentation. And there's quite a bit of diversity when you look at all the organisms. Remember, many of these prokaryotes are growing, living, North Pole to South Pole, every place in between. So yeah, we know the sugars, but you can also see amino acids, fatty acids. Remember the purines, pyrimidines, that's nucleic acids. Some aromatic compounds can get broken down and fed into some metabolic pathway in some way so that you can get at excited electrons. You can get at some substrate level phosphorylation to make ATP. That's where that whole acetate in the last section comes into play. You can take fatty acids, cut 2, allows for some substrate level phosphorylation.